So I just want to echo Chris's comments in terms of this idea that we're really excited to have everybody here. We kind of came up with this idea more generally speaking, and as I really started to think about all of you and the things that you do, I got more and more excited about having everybody here together. And so I'm really looking forward to the next 2 days of being an opportunity for us to really communicate about the work we do, whether it be in the microbiome or other areas associated with health disparities, and really get a discussion going, because I think there's a lot of work that's going on in the microbiome world right now that's highly applicable, obviously, since you're all here. But oftentimes it doesn't get attached to that formal title of being associated with health disparities. And so I think it's a really nice opportunity to have people that sometimes aren't thinking either about the microbiome or about health disparities directly really start to interact and think about that. And so, as Chris said, I'm going to provide our first talk here. A lot of my work in the past has been on non-human primates. And just to kind of forewarn you, I said I was going to talk about my comparative work between non-human primates and humans, and I'm happy to talk with anybody about that in terms of the kind of the cross-species comparisons I've been doing. But I decided that that was more related to human health and physiology and not necessarily health disparities. And so I'm really going to talk about my work with non-human primates and how it's informed some of the things I'm thinking about in terms of moving into human work as well. So still giving this kind of evolutionary context, but I'm not necessarily going to tell you how directly similar humans and non-human primates are, although I'm happy to talk about that with anybody that wants to. So, we all know I don't have to tell this audience, right, but you have to start with your slide about all the effects the gut microbiota has on hosts, right? So we can put a lot of things into all of these categories, but we know gut microbes affect host nutrition, immune function, mental state in terms of the gut-brain interaction, and so as a result, there's been a lot of research and interest in linking the gut microbiota to different types of health statuses. Okay. And we know that the gut microbiota is implicated in a whole host of diseases, chronic or otherwise. I've listed a few here, but we know that it's involved in many, many more, at least in terms of being associated with these things. And we also know that a lot of these same conditions, basically all of them, can be associated with health disparities. These are things that we see come up when we're talking about health disparities and differences between populations and their health outcomes, et cetera. And so really the obvious question, and the reason we're all here today, is trying to understand how important are gut microbes for understanding patterns of health inequality across populations. Can we use this as another tool to look through the lens of health disparities and try to understand kind of where these differences are coming from? And so, like I said today, I'm going to give you a little bit of my perspective on it in terms of an evolutionary context. I tend to focus most of my work on trying to understand host nutrition and energetics and the effects of gut microbes on that. And so I'll really kind of focus on that context and the work that I've done with non-human primates and how that informs it. In particular, I want to focus on metabolic disease, so obesity. So this is just an example figure of looking at prevalence of obesity in different populations. So this is children and adolescents aged 2 to 19 years, right? We're looking at boys versus girls, and you can see different populations have different rates of obesity, and also in terms of race and ethnicity, and also in terms of things like socioeconomic status, right? So there are health disparities in how obesity presents itself, at least in patterns across populations. And the obesity work that came out 10 plus years ago now is really what started to get me interested in the gut microbiome, right? So Peter Turnbow and everybody else on this paper published this in 2006, right? It was this really transformative idea that gut microbes actually had a causative effect in terms of determining obesity, right? So a certain type of gut microbiota leads to an obese phenotype, right? And they associated this with the potential for the gut microbes that you find in the obese phenotype to produce more short-chain fatty acids, right? And we know that short-chain fatty acids can be absorbed by hosts and used as energy, and then if you're not using that energy actively, presumably you have to store it, and that contributes to weight gain, okay? And so this was a really interesting relationship to think about, and people really jumped on it in terms of thinking about medicine and therapies and interventions and kind of clinical studies. And I saw this research and I thought it was interesting, but it also made me stop for a second and say, well, that's all well and good, and it could be bad if you're not using that energy, but what if you needed that energy, right? What if you were in a situation where your battery was low, so to speak? Having microbes that are potentially more efficient at producing that energy for the host could then be a good thing. That would be evolutionarily advantageous. And there are a few different scenarios where you could think about this happening. One would be food shortages, so whether those are short-term or long-term food shortages, right? You don't have enough to eat. If your gut microbes can somehow shift and get more out of what you're eating and provide you with extra energy that you wouldn't otherwise be getting, that would be a good thing, right? And you wouldn't necessarily be storing fat, but then you would be getting through this lean season, so to speak, okay? And then the other areas are associated with things like life history processes, right? So pregnancy and lactation in females, and then growth in juveniles, right? These are periods during which we have higher metabolic demands. And so again, if you have a gut microbial community that can kind of kick in and provide extra energy during those periods, it's potentially an advantageous sort of thing. And so this is kind of where I started to think about it in terms of what I wanted to study in terms of the microbiome and contribute, using the same sorts of ideas and processes, but really looking at them from this different perspective. And this is how I started working with non-human primates, because a lot of the populations, especially at that time that were being studied, I wouldn't say were necessarily being nutritionally challenged, right, in terms of either food shortages or those life history demands. So populations are being studied with people like me, right? If I'm hungry, I walk down the store, I get more food, and it's not that big a deal. And so microbes arguably could be less important nutritionally in those contexts. But when you have wild non-human primates, you actually are in these sorts of situations and scenarios where you go through things like food shortages seasonally, and where females may not be able to just eat more if they're reproductively active. And so I started studying howler monkeys in the wild in southeastern Mexico. And I was really interested in howler monkeys because they have a very variable diet over seasons. So during some seasons, they'll eat all fruit. And then during the season when there's less fruit around, they'll switch to a diet of almost all leaves, which are very high in fiber, secondary metabolites, hard to digest. We think of that as being a lower energy food resource. And for a long time, people had said, well, these howler monkeys probably are just less active, and then they also maybe rely on gut microbes for that, but we didn't have the tools to actually look at it. So I wanted to go out and look at it. This particular species of howler monkey also has very cohesive social groups, and so when a female is reproducing, it's not necessarily that she just goes off and eats something different. Basically, the whole group is in a tree at the same time eating the same stuff. And so again, there's limits in terms of behavior with regard to how females might be able to respond to things like pregnancy and lactation, and the same would go for juveniles. So I followed these monkeys around, chased them around in the forest for a year, and picked up fecal samples every 2 weeks, and was also recording their diet, and tried to record reproductive state actually, but they all reproduced at the same time, which was a bummer for me. So season and reproductive state were confounded, but I did have males, females, and juveniles. And I wanted to look at if there was maybe this sort of compensation that might be happening during periods of low food intake. And what I can tell you is that yes, it looks like there is that sort of process happening, right? So in our ordination plot here, each of these points is one howler monkey, and this is the average for a season. And the same howler monkeys were sampled across all 3 seasons. And the different colors are the different seasons, and you can see a very clear clustering plot. So different gut microbial community across different seasons. And what was interesting is that while I was in the field, there were variations in how much fruit and leaves, but actually as I went through the year, the estimated energy intake in terms of easily metabolizable energy that these howler monkeys were consuming decreased. And as it decreased, we saw shifts in the relative abundances of certain microbial taxa, right? And I don't have the functional data for these microbes here, but what's interesting is that both of these groups we know are associated with things like carbohydrate breakdown. And so there may be something going on here in terms of the types of whether these howler monkeys are eating a lot of fiber in their diets or not, and how much energy is getting pulled out of that. What's also interesting is that as this happened, as this energy intake decreased across seasons, the amount of short-chain fatty acids, those molecules that can be used for energy, actually increased in the fecal material. So again, it looked like as the monkeys were consuming less energy over time, the gut microbial community is potentially producing more short-chain fatty acids. And their activity budgets basically stayed the same across seasons. There are no differences, there's no seasonality in breeding, et cetera. And so there's potential here that these gut microbes may actually be helping these howler monkeys compensate. In terms of looking across age and sex classes, the patterns were much more subtle. This is a relatively small sample size. There's only so many monkeys you can chase around at one time. But we did see differences in juveniles compared to adults and in adult females compared to adult males. And a lot of what we saw were increased relative abundances or increased prevalence of things like firmicutes, which were one of those phyla that the Turnbow paper pulled out in terms of potentially being more efficient at producing short-chain fatty acids, right? Now, we can argue about whether they're actually more efficient or not, but it's interesting that we do see this pattern. Adult females were also characterized by lactococcus. This is related to microbes that could produce folic acid, and so this is something interesting to think about as well that I'd like to go back and look at in more detail, kind of pull genomes out and see if there's actually potential for folic acid production here. So there may be some of this compensation happening here. So that's all well and good, and that there may be there's evidence for these potentially beneficial evolutionary relationships between hosts and gut microbes. But then the question becomes, do these apparently beneficial relationships ever break down? And I think this is where it becomes more relevant to thinking about the context of human health disparities, et cetera. So what do we see in non-human primates? Well, I saw that across seasons as diet shifted, so did the gut microbiome. And when we look across habitats, the gut microbiome of these howler monkeys also shifts. So again, this is just taxonomic composition. You can see clustering. I sampled monkeys in a continuous forest that was either evergreen or semi-deciduous, as well in a forest fragment, and then in captivity. And you can see there's different microbial communities within the same species depending on the habitat in which you sampled. What's interesting is that in this case, it doesn't look like there's necessarily a compensation, necessarily a kind of a good thing going on. What you get is that as the habitat quality is decreased, generally speaking, you get a reduction in the diversity of the gut microbial community. And we tend to associate, of course, increased diversity with things like resilience, functional redundancy, et cetera. And so this is a potentially bad thing for the health of the howler monkeys, right, in terms of their gut microbial communities. And when we went in and did some qPCR and pulled out genes associated with some specific microbes, we also saw things like reductions in microbes like Butyrivibrio, these short-chain fatty acid producers. So nutritionally, these howler monkeys that are in these kind of lower quality environments and that their diet is extremely altered are also seeing a reduction in this potential for short-chain fatty acid production, okay? And the same pattern holds if we go out and pull out the actual genes for like butyrate production, one of these short-chain fatty acids. And things that can produce toxic byproducts that are associated with things like colon cancer and gut inflammation like disulfovibrio. To some extent, you're seeing more of those in these more degraded habitats. And so these data suggest that there's a limit to this compensation, right? And if you're actually in a certain type of habitat, you may be exposed to different types of microbes, presumably. I think this is associated with their diet. There's very different diets in these places. And the gut microbial community is going to shift, and there may actually be health consequences for those sorts of relationships, okay? And so this may be a place where you see these relationships break down. And in fact, from another study I did where we actually sampled howler monkeys in fragmented rainforests, several species of howler monkeys, you see less response to seasonal changes in diet in howler monkeys that live in a fragmented rainforest compared to those that live in a continuous rainforest, right? So there may be this reduced capacity for this kind of flexibility of the microbiome if you're starting out at a point that's kind of more quote-unquote degraded. The extreme case, of course, is captivity in primates in terms of the diet being extremely different and environmental exposure, which I'll talk about in a second. But a lot of data sets that I've had the opportunity to work with lately have comparative data from captive and wild populations. So I just showed you data from howler monkeys that suggest that captivity kind of does a number on the gut microbiota. These are data from Asian colobines, so things like snub-nosed monkeys and Duke langurs. They have a sacculated foregut that's analogous to a rumen in a cow. There's differences in terms of what the gut microbiota looks like in captivity in the wild. These are data that an undergraduate honors student of mine produced, looking at 5 different species of primates in the Lincoln Park Zoo compared to data I had from the wild, so chimpanzees, gorillas, colobus monkeys, howler monkeys, and red-tailed monkeys, so apes, old world monkeys, and new world monkeys. And you can see, again, very distinctly, there's differences in the gut microbiota of captive and wild animals. This study was published a couple years ago, again, looking at the same sort of thing. These are those Asian colobines, howler monkeys again. These are individuals that are in captivity. These are kind of semi-wild, and then, wild, semi-wild, and then captivity compared to human data. And the message here was that captivity humanizes the primate microbiome, okay? In that the gut microbiota of these primates starts to look more like what you would expect from a human than what would you expect from a non-human primate. And we see the same sort of pattern in diversity that I showed you in my own howler monkey data, in that these captive and semi-captive individuals have lower microbial diversity than wild individuals. The question becomes, does this matter? Does it matter if you have a different gut microbial community, right? You're in a different environment, your gut microbial community is different. Maybe that's good, right? Maybe it's actually this evolutionary benefit. We know that non-human primates have a lot of issues with gastrointestinal disease in captivity. And so we think that these shifts may not actually be a good thing in terms of the rest of the health of the animals. And this is particularly true for some of these non-human primates that have more specialized diet, like howler monkeys and colobines that consume a lot of leaves and really rely on gut microbes to break down fiber. But in a lot of cases, including in my wild data, we don't actually have the health data to look at this. And so this is a big gap where we need to start to fill things in. I will tell you I did get access to data from the San Diego Zoo, where they were doing necropsies on individuals that had passed away for various reasons. And these, again, are colobines, so they have that specialized foregut. But they're looking at whether the San Diego Zoo was interested in whether these individuals that had died from gastrointestinal causes had a distinct gut microbiota compared to the rest. And in particular, they had issues with their Duke langurs getting sick. You can see almost all of these teal dots are large, which means they were unhealthy. Small dots are healthy individuals, healthy at the time of death in terms of their GI tract. And we did find evidence that the Dukes had a different type of gut microbial community, and when they were sick, it shifted even more, right? And so again, there's evidence that these shifts in the microbial community are associated with disease. Now, it's correlative, right? And we're not getting at causation necessarily here, but there's this idea that having a gut microbial community, potentially in a captive context, depending on the primate you are, may actually set you up for disease in that environment, right? There may be sort of a mismatch in terms of the microbes you have and then your ability to survive in that environment. And then the last thing I wanted to show you in terms of captivity is a lot of times these studies associate the changes in the gut microbiota with changes in diet, okay? And here we're really not just talking about diet. So this is a study where we have macaques from Puerto Rico that are either semi-wild on Cayo Santiago or in actual captivity on the mainland, and they're being fed the same diet. So in Cayo Santiago, the macaques are actually provisioned. They're fed the same monkey chow that the macaques in Sabana Seca in captivity are being fed. And so there's really not a huge difference in diet, and the difference here is really environmental exposures, okay? And again, you can see that here are our Cayo Santiago individuals, and here are our Sabana Seca individuals. So even though their diet is the same, being in captivity versus not leads to differences in the gut microbiota. And so this idea of environmental exposures and the effect of that on the gut microbiota is also important in the sense that it's not just diet that's driving it. There are other things about the environmental context that it can actually influence this baseline composition. And so the question becomes, are these processes transferable to humans, right? What does this mean in terms of humans? Do we see the same sorts of evolutionary relationships in terms of potential benefits in humans? And do we see the same sorts of environmental effects on the gut microbiota that may actually play into your propensity to become sick and may be associated with things like health disparities? I would say in terms of the evolutionary side of things, probably. I would be surprised if the same processes weren't at work. But I would argue that we need more data. So things that I looked at, like seasonal variation in food availability in non-human primates, we actually don't know a whole lot about in humans. There's basically been a couple studies, one of which Emily wrote, that have looked at this. And it looks like there is an effect. There are differences. But again, the functional consequences of those differences are something we need to really dig into more. And there's a lot of studies on pregnancy and reproduction, and there may be a few more out there that I've missed or things that are being done. But a lot of them, again, focus on disease, and they're not necessarily looking at the normal course of pregnancy in terms of this idea of compensation. Although, again, the one that does suggest the same thing happens, that there's shifts in the gut microbiota that actually might help mom in terms of energetic production. We also know that context matters for human populations, right? So I fit as many graphs on this slide as we can, but we know there's a zillion papers that look at the quote-unquote Western versus non-Western gut microbiota, right? So looking at people living in more industrialized places with distinct diets compared to those that don't. And we see the same sorts of patterns that we see in response to habitat in non-human primates. And a lot of these patterns are associated with similar things that we see with like captivity in non-human primates, reductions in diversity, shifts in certain types of gut microbes. And so you could argue that this, again, is kind of a similar situation in that you're now getting different either environmental inputs or that differences in diet actually make people start at a different baseline in terms of their gut microbial community. And we, again, know that people living in industrialized countries have these different gut microbial communities. Those are associated with these chronic diseases. There's more of those chronic diseases in these industrialized populations. So similar relationships that we're seeing in the non-human primates. And so this, again, is this kind of parallel situation in humans as it is in non-human primates. And I think this is where we can really start to think about health disparities, although these are on a much larger scale, right? And I think we need to start to look within populations at kind of a more micro scale, right? Obviously, all the people living in the industrialized nations don't have the same access to food, have the same environmental exposure to microbes, et cetera. And I'm looking forward to hearing everybody here talk about that more. The last thing I wanted to bring up is this question about, I've talked about, environmental exposure, your diet, kind of altering your start point, your environment changing your start point, and then that having negative impacts on health. But I want to go back to this idea about these kind of mismatched situations, so to say. So really go back to where I started in terms of the obesity research, right? In terms of looking at, we think of this as a bad thing. You're not necessarily using that energy, right? But what if you needed that energy, right? What if you got set up with a microbial community that was good for the environment you were in because it was an environment where you had low access to energy resources, but then your environment changes, right? And you have the microbes that are set up for this sort of situation, but you're not in that environment anymore. And so this relates back to kind of global patterns in metabolic disease and obesity. So we know this is becoming a big problem, especially in particular countries. And it's starting, the rates of obesity, there are nations where rates of obesity are already high, places like the United States, right? But there are also a number of countries who are actually seeing increased rates of obesity, right? Where these are projected rates of obesity. And you can see in a lot of places that are considered to be developing are becoming kind of more industrialized, the political economy is shifting. Basically, we're expected to see larger increases in the rates of obesity than we are in these nations where we're already suffering from high rates of obesity. So in terms of this mismatch scenario and these shifts in the way these economies are working in these places, I'm really interested in understanding, and these are obviously extremes, right? But what happens when you go from people living with these sort of nutritional limitations potentially to people living in an environment where the nutritional, I wouldn't necessarily say the nutrition is good, but in terms of energy anyhow, maybe potentially less limited based on the way that the economy is working and the type of lifestyles that people are living. And you could actually get a scenario in which your gut microbiota is set up for something like this, but now you're living in something like this. And does that actually contribute to the emergence of things like metabolic disease and the rapid rates at which we're seeing this happening in these countries that are undergoing economic transitions? So some of the major questions I'm interested in this context, and this is a study that I'm launching in collaboration with Chris and Tom McDade at Northwestern. We're really interested in whether a maternal history of undernutrition, so not having access to enough nutritional resources, particularly in the form of things like calories, energy, in early life, does that lead to a more energy efficient, quote unquote, gut microbiota in adulthood? And can these microbial traits be passed on to infants and then affect their health outcomes? And particularly in this context of that nutritional environment shifting. And so this is a study that we're doing in metropolitan Cebu in the Philippines. This part, the Philippines has undergone this economic transition relatively recently. People's diets have shifted over the past couple generations as the economy has shifted. And what's really cool is that there's the Cebu Longitudinal Health and Nutrition Survey, which Tom and Chris have been involved with for a number of years. And we basically have data from mothers and their offspring that were collected starting in 1983. So over 30 years of data describing the health of mothers when they were pregnant and then tracking them out into the present. And then their offspring, we have data from them basically starting from when they were in utero all the way until they're adults, okay? And these adults are now in their 30s and they're starting to have offspring of their own. And so while we don't have fecal samples that are banked, we know a lot about the nutritional status, let's say, of these offspring during their early life. And we can use that to ask questions about what happens to the gut microbiota in adulthood and then what gets passed on to offspring. And so this is kind of the study design we're going for and trying to think about this. And again, this is an extreme. Our population is probably going to be more of a gradient, but just in terms of conceptual outlining here, we're looking at mothers that either had limited access to nutritional resources during their early childhood versus not, so versus kind of this normal exposure, normal nutrition. And we want to look at if their gut microbial communities are different, both in terms of composition and function when they're adults during pregnancy. We're going to sample them during the third trimester of pregnancy in terms of fecal samples to look at the microbiome, as well as get some physiological data in terms of looking at things like BMI, things like metabolic state to some extent, and then also looking at their diet. And then when babies are born, we're going to sample them and compare their microbial communities to moms to look at this transfer, and also compare the microbial communities between babies that had moms with this history of undernutrition during early life versus moms that had adequate nutrition during early life. And we want to see if their microbial communities are similar or different, both in terms of that composition and that function. And then the idea is to follow babies out as they get older, right? And so looking to some extent at how the microbiome develops in babies, but continuing to do these comparisons in terms of what are the differences between these babies whose moms had different nutritional histories, okay? And how much of that signal do babies maintain as they get older? And also looking at the physiological development of these infants, right? So do you see, for example, infants that were born to moms that were undernourished, do these infants actually gain weight faster? Do we see risk factors for things like metabolic disease because moms potentially had this gut microbiota that was more efficient for that kind of reduced environment compared to babies that have moms that didn't have that sort of nutritional challenge? Do we see less signs of this sort of metabolic risk here? So hopefully I'll have some data to talk to you about that soon. We're actually receiving our first samples probably in the next month or two. They've started to be collected. And I kind of want to leave it at that and let the rest of you talk about what you see as kind of interesting areas to think about health disparities and things that are being done and that can be done. But kind of my final thoughts in terms of my own take-home message is that to understand what is quote-unquote wrong with the microbiome when there are health issues, we need to both understand what was evolutionary quote-unquote right? Right and wrong aren't really good evolutionary terms, but throw them in there anyway. And then we need to understand what the perturbations are that have actually altered the system, right? Do we have these sorts of evolutionary mismatches that are happening, right? Is the response of the microbiome to certain things actually an evolutionarily advantageous response, but we're just playing it out in these environments where it's really not matched there and it becomes a problem, right? Obviously, studies of human populations are critical. There's a reason everybody else here studies humans. But I think that we can also not discount non-human primates in terms of their ability to give us an even broader context and try to kind of understand how these host-gut microbiome relationships may have evolved in selective environments and what may be similar or different in terms of the human situation, both in terms of using non-human primates as actual models and then setting them up as contrasts and using them in a comparative sense. So that's all I have. It's the funding associated with some of these studies. And I don't know that I have an actual questions slide, but I'm happy to take questions. If I have time. Yes. Sorry. Anoop, and then I'll get Lipe.
So I have a question about the adjustment to obesity. So the slide you showed with figure increasing obesity in areas that are calorically challenged, let's say. And then the slide pictures of mom females. And I'm trying to think about what the I'm approaching this from the obesity literature. So I understand that there's some adjustment. Like I'm in this low calorie environment, all of a sudden that changes, then I have the wrong microbiota. But the question is how long it takes to adapt that and adjust my microbiota for richer environment. It could be one generation, which is just a dislocation for one generation to obesity. And the reason why I ask that is because at some point, say in the US, you must have had a change from we knew that in the 1800s there was a caloric scarcity. Now there isn't. Obviously, it was a big obesity boom. It was more of a gradual change. And the reason there's a big obesity boom is because there's more dramatic change in developed countries. But our history would suggest that maybe down the road, you have more moderation in the or change in microbiota so that there'd be less obesity down the road. Does that make sense?
Yeah, it does. I think my insufficient answer is to some extent we don't know. So we know that the composition of the gut microbiota can change relatively quickly if your diet change is dramatic enough. And then also, it's interesting, and I don't know, maybe Megan can speak to this a little bit, but this idea of microbes being passed on from mom to offspring, presumably there's going to be a certain number of generations where that might start to peter out. But I don't even know if we have a great idea right now of how much of the signal from infants gets carried into adulthood. And that would be my question is what is kind of the effect, what is the strength of that in terms of what gets carried on to adulthood, and then how fast can you see kind of that be eliminated and change? And how important are those microbes in determining these phenotypes also, right? Are the microbes that are getting passed on, presumably the ones that are getting passed on should be important for determining phenotypes. That's why they would be passed on in terms of being evolutionarily advantageous. But I don't know that we know enough about the signal and how it's maintained. I don't know if anybody else has thoughts on that. See. Yeah.
I had a related sort of question. So in the study you described, where you're going to look at the moms who are undernourished versus normally and the babies and looking at their microbiome, are you also going to do epigenetics in that cohort? Because I think of the microbiome as more dynamic, you can adjust really quickly, whereas things like your genome in the very evolutionary sense or your epigenome, like kind of in between, might be more static. So you really need to layer those two things.
Yeah, I know there have been epigenetics done on this population. Chris, do you want to do epigenetics? Are we going to do epigenetics in the Cebu study on this cohort?
Yeah, so we've measured genome-wide methylation in about 400 women and 100 men. It's actually Tom McDade who just arrived, who spearheaded that. And so we've done a fair amount of that, but mostly looking at effects on immunity because it's immune cells. So it's whether or not this is going to tell us something about something like obesity, I'm not sure.
Yeah. Yiping, did you?
Yes. So I think the contribution of short-chain fatty acids produced by gut bacteria to obesity is really still an open question. And so generally the field, nutritional field has a consensus that about 10% of the calorie the daily intake are probably from short-chain fatty acids. But if you look at the body weight growth, when people start to develop obesity, that amount of calorie cannot explain. Probably starchy food and high calorie intake from starchy food may be a very important reason, because Jeff Gordon's group pioneered to show that gut microbiota colonization of the germ-free gut with normal microbiota can promote genes in the liver for converting glucose into triglyceride to a very high level. So that means gut microbiota actually helping facilitate the transformation of dietary intake in the form of glucose into fat.
Yes, and I didn't get into all of the mechanisms here, but I don't think it's just short-chain fatty acids, the amount of energy. I actually think I'm still interested in the short-chain fatty acids, but also, just like you're talking about, how they actually metabolically program the host, and what different types of short-chain fatty acids, how they affect host metabolic programming distinctly, and associated with how much fiber is in your diet versus not. Yeah.
So short-chain fatty acids can be from all kinds of organic compound fermentation, not just carbohydrates. They can also ferment proteins, fats, and choline, all kinds of organic compounds, and produce short-chain fatty acid. So I think the story is much more complicated than it looks like.
Yeah, no, absolutely. It always is. Laura, and then
Okay, so your maternal study is very, very interesting, and I wanted to know if you were including or excluding mothers that deliver by C-section, or if that could be a control for mothers that would have less transmission of microbes to those infants.
it would be a great control in terms of the transmission. We are not. Sorry, go ahead.
So is it an exclusion criteria at this moment?
It's not. We have almost no exclusion criteria at this point, because they're in their 30s. We're starting to kind of get toward the end of peak fertility in this population, and so we're basically taking everybody we can get into the study. And so in terms of some of the analyses, we may or may not exclude, but there probably will be some C-section. I don't think the rate of C-sections is very high in this population, though.
So that would have been back in 1983. Very
Okay. And then
But
also
now,
considering antibiotics in that population to disrupt the normal transmission. That might also be an interesting control if you don't see if you see a loss of transmission with associated with antibiotics. That could be very interesting as well.
Yeah, it would be really interesting. And we are asking moms about their antibiotic intake and also infant antibiotic intake, but again, not excluding anybody. So depending on sample size, we should be able to address that. Ross?
So I have a question actually linked to Lupin's question too. Have experiments been done where, for example, the lean microbiota have been supplemented with short-chain fatty acids or something? Seems like, and then, I mean, you'd expect mice to get obese if that was the cause.
Yeah, Lupin may know more. I don't know about lean individuals. I feel like I've heard of the opposite being done. So when there's metabolic disease, being supplemented with certain short-chain fatties like butyrate. And my understanding is that doesn't have a huge effect if you just, but you're also giving it to them differently, right? You're giving it intravenously. It's not going through the gut necessarily. I don't know about lean individuals being given short-chain fatty acids.
Interesting question. I think a Yale group, they published a Nature cover story, and the conclusion is acetic acid is driving obesity. But actually, the critical evidence is in intragastric infusion of acetic acid. I think that's wrong, because you never have opportunity to get such amount of acetic acid in your gastric, in your stomach. It's the lower cut. If they do intracolonic infusion, I think they would get opposite conclusion.
So they want to be able to know.
Yeah.
Because in general, this was a question about types of experiments that you can do, targeted experiments rather than descriptive measurements. And the other one that I had in mind after you presented your work was would it be, ethically feasible to take mice, make them pregnant or get them pregnant, in some way, and then put them in some kind of a limited environment, you know, and then feed them either with the lean or the obese microbiota and see whether the ones with the obese microbiota would predict get healthier babies in a situation of food shortage. It seems like a simple experiment, but
Yeah.
nature is allowed. But,
Yes, potentially. And I am starting to do some mouse work along kind of a similar vein, but it's more human, non-human primate comparative, but also looking at short-chain fatty acids. You could do that. My only hesitation with the mouse work is that even just body size, the metabolism of a mouse is so different than that of a human.
the point
But
is
yeah.
that for the mouse, we already know you can get them obese by introducing this microbiota.
Yes.
So that's a good starting point.
Yeah, no, those would be really interesting experiments, absolutely.
And there's a tiny little other question. Why is this obesity so low in Australia?
Low?
Map. Yeah.
It's high. It's very high.
Oh, the new map said very, very low numbers.
That was the 2007 paper, wasn't it?
Oh, yeah, it is low.
The rest of the world, it's
But if you
more.
go back to the other map,
Yeah, the other map, yeah.
It's very high.
Oh, what is this other map showing then?
This is showing this is published in 2007. So this is data from the year 2000 and then projected in 2030.
What data? Obesity rate? Because that.
Yeah. Yes,
Okay, then-
percent
what's-
of the population
Okay,
considered.
something is unexplained.
And these are from 2014, so this is 7 years later. And when I was looking through for figures of this, everything Australia had a pretty sharp increase. So I wouldn't be surprised if it's changed a great deal since that 2000 data, year 2000.
Okay, maybe one more question.
Yeah.
About the theory of having an energy efficient microbiota and kinds of proteins. If I remember correctly, there was some studies that used to transplants between lean and healthy donors and obesity ones. And usually the lean ones receive the stool from healthy donors and they lose weight. So even as opposed to the theory in times of the freedom starts to be developed as energy efficient.
Would it oppose that? Why would it?
Because these you know, you suppose that the lean and healthy donors will eat less food than obese people. It's transferred into the obese people and they lose weight. So it seems that this microbiota is not necessarily more efficient in processing food.
Which study you refer to? I don't see such study. I know one study. So they did either self-transplantation as control or donors from healthy donors. Then the treatment group didn't show weight loss, but they show temporary improvement of peripheral insulin sensitivity. Yeah, but no weight loss. No weight loss. I think we would. I think we need to be careful when we look at different publications. Like Professor Gordon's group, they did microbiota transplantation from obese mice to either from obese mice or from lean mice, and then you see increase of adiposity, increase of fat deposition, but the body weight is no difference. It's only the body fat, the body composition change. So it's not transfer of obesity. It's transfer of a little bit more adiposity, a little bit more fat. So then, but in the news medium, they show exactly the picture. So this is a small, and that one is very, sometimes it could be misleading.
Right. Yeah, no, I think the metabolic programming angle of this is the interesting angle, for sure. And
Thanks, Katie.
thanks.